Okay, welcome back after the lunch break. And we have an exciting list of speakers to keep you all awake as you begin to digest that lunch. And we're going to start off with Alison Cameron, who's going to be talking about deforestation and ecosystem services from soil. Thanks, Alison.
Thank you very much. So I'm working with the Pages project in Madagascar. So this talk follows on very much from Mafika's talk. I'll be presenting the data that's being collected by his work package, by the hydrological work package. So our project is called Can Paying for Global Ecosystem Services Alleviate Poverty? And we have a large team, as you'd expect for an ESPA project. So really all things have moved around on the screen a bit, but this talk really focuses on data from the biophysical work packages. There are socioeconomic work packages attached to the project, but I'm a natural scientist, and at the moment I'm busy trying to synthesize the results of the biodiversity, carbon, and hydrology work packages, and then that will be synthesized with the socioeconomic data at a slightly later date. But even with just this subset of our project, we have a really large list of authors that I have to thank for their data and for participation in this to produce this talk. The overall goal of our project is to influence the development and implementation of ecosystem service payments, international payments, in the interest of poverty alleviation. So to try to make sure things are a bit more sensible. For example, RED would be a good example of a payment scheme, and is specifically one that we are working on in the project. So in our context, what we're interested in is how natural forest and perhaps different states of degraded forest are delivering hydrological services to local people in Madagascar. And I think that Mafika has done a very good job about talking about these local hydrological benefits, but of course we're also interested in the global benefits in terms of climate change mitigation and climate regulation. And I'm a biodiversity scientist, so I'm interested in how the biodiversity and the functioning of these forest systems interacts and creates these services. Now, this explains the project as a whole. And so we have a landscape in Madagascar which is being degraded from natural forest through various stages of slash and burn agriculture. So it's a very patchy, heterogeneous landscape. And there's a gradation going from closed canopy forest through these states of degradation to agricultural land. And I'm focusing on hydrology, biodiversity, and carbon. Other groups are looking more at institutional filters and how much money gets to who, and whether project-affected people are correctly identified, et cetera, but that's not what I'm going to talk about today. I focus on these biophysical components. Now, the background to this, Steiger published a lovely paper in 2007 which has simplified our life slightly, where they interviewed people about their land use practices and characterized this slash and burn agricultural system, which is called a TAVI system in Madagascar. Probably got different names all around the world. And ultimately, it's a cyclical system. So people start off with closed canopy forest, they cut it and burn it, and they grow their first crop, and then they let it recover, and then they cut and burn it, and they grow their next crop. And if you have a long enough fallow cycle, in theory, you could maintain the system in a steady state between agricultural productivity and shrub fallow, or between tree fallow and agricultural productivity. In Madagascar, unfortunately, people don't leave long enough. There's a lot of population pressure. So we have an accelerated degradation now where we're calling this business as usual scenario here, where people are slashing and burning too frequently. And the land is being driven down this side of the trajectory to what is known in Madagascar as dead land, or tani mati. And that speaks for itself. This is really beyond agricultural use. It is possible for that land to recover, but it's a slow process. And there's really been very little intervention to try to accelerate this. It will happen naturally, but there's some projects have attempted restoration work. So we've been interested in characterizing the ecosystem services on both sides here, so comparing a new shrub savoca, which has been recently deforested versus a recovering shrub sapoca. Are they the same or are they different? And also looking at how effective restoration is. So that's the bigger picture of what we want to do. Now, if you looked at this, you would think, hang on, what's going on? This is rice productivity. Now, we're not talking wet rice like Mafika showed pictures of here. This is hill rice productivity, dry rice, where people plant exactly where they felled the trees. And you can see that rice productivity increases. It's not actually high in the first cycle. So there's an incentive to keep clearing until you have shrub sapoca. First of all, it's easier to keep managing shrub sapoca. You're not having to deal with trees every time you want to clear and grow your crop. But secondly, actually, the peak productivity comes here. But you can see these declines. And so eventually, there's really no point in planting crops anymore. You've got to this dead state. So really, it would make sense to try to maintain a sort of equilibrium of a lot of the landscape being in shrub fallow. Because there's been some work done to characterize the species that characterize each of these different stages. So this was useful foundation work for us. So you'd expect a lot of land to be in this condition optimally, because it'd be producing optimal yields. Now, these are the the SDGs, and the ones that have been identified by the Institute for Advanced Sustainability Studies as relating to soil sustainability. Now, we are not really working too much on health metrics or gender equality, and we're not really touching on cities, but essentially our project does focus on all these other ones, so we're covering about 6 of the SDGs really in this project. Our study sites, as Mafika showed, these are the social science study sites, and these are, the biophysical study sites. So we've got 3 zones of interest spanning areas which are more rapidly deforested, perhaps have a more stable forest boundary, slight cultural differences, altitudinal differences. In this presentation, I'm only presenting about half of the data because that's all that's really been worked up in the laboratory so far. Our field teams really have just finished field work. So we have currently 21 sites processed for biodiversity data from Zone of Interest 2, sorry, 11 out of 21. Zone of Interest 3, we've done all the sites, and we haven't touched on Zone of Interest 4 data yet. So this is kind of a preview. It's not going to be published in this form. We'll have a bigger data set when we get the full works. This gives a cartoon illustration of what we've managed to sample. So far, we've got 4 closed canopy sites, 2 tree sapoca, 5 shrub sapoca, and 4 dead land or degraded land sites. And in addition to that, we've got 4 intervention sites where there's been reforestation. There's one site up here that we thought was closed canopy, but it turns out it's been an intervention site. It was cleared by logging, selective logging, not completely cleared and then enriched. But we're currently just lumping that in with closed canopy because it was structurally indistinguishable. So this is what I'll present, but there will be a lot more than this by the end of the year. So these are our field teams in action, and if you can't get enough money out of ESPA, then you can make Paul dig holes for you. And so there is a lot of hole digging. It's a common theme, actually, across all of this. So these are infiltration sites for hydrology, and then they pour water in here and look at how long it takes for the water to go down the column. Carbon sampling involves, we have above and below ground carbon sampling. If anybody wants to talk more about carbon sampling, Narivo, who led all of that, is here and giving a poster. So please quiz her about the details of this. And then biodiversity sampling. Now, I'm focusing in this talk only on soil biodiversity, but we've sampled amphibians and reptiles and looked at a couple of above ground groups as well as below ground groups. And we've sampled invertebrates above and below ground. This was one of our biggest challenges, and it sounds so simple, to get all these people into the same site to collect the same data, comparable data. And then we realized that the trouble is that we would all destroy each other's sampling by trampling over it. So we had to put people in different stages and very carefully spatially map out our sampling sites with our biodiversity sampling slightly offset from the carbon sampling, because the carbon sampling was so destructive. And the hydrology sampling followed the biodiversity sampling along this line. So the hydrology samples were literally in the same hole as our soil biodiversity sampling, and the carbon was just offset by a few meters. And in general, we were pretty successful at getting everybody into the same sites. We've collected a vast array of variables from each different work package. So this is just a list of hydrology ones, and I don't want to go into them, but what I wanted to point out was there's so many different things you can correlate. So you could look at one hydrological variable against one carbon variable or one biodiversity variable. And of course, you could fall into that statistical trap of just making so many correlations, you eventually get some significant ones. Now, we do find across a lot of these variables declining hydrological functioning, but what's actually quite optimistic is that the reforestation sites are performing a lot better than people gave them credit for. The biodiversity community were very sort of despondent about how long it would take for these sites to do anything for biodiversity. And we're discovering that sites that are only 6 years old in terms of their restoration history actually have a significant improvement from the degraded sites and the fallow sites. So we've been having to use, instead of just making lots of comparisons between individual variables, we're having to use multivariate statistics to try to look for patterns across these data sets. And I don't know how simplistic to be about this, because some people, the minute you mention multivariate statistics, they switch off and they're scared of it. But ultimately what we're doing is creating these distance matrices between plots to look at the degree of similarity between samples. So we create a similarity matrix for each data set. So we'd have one matrix like this for biodiversity, and it summarizes all the variability in all of the biodiversity variables. And we create another matrix for hydrology and another matrix for carbon. And so this is just zooming in slightly. So you can see that we would compare the similarity of this reforested site with this closed canopy site, and this dead land site with that closed canopy site. So every site gets compared against every other site. I'll start with just very simple principal components analysis, which is where you take those distances, similarity distances, and you can plot them out on 2 dimensions on a graph. So 2 dimensions is a limited way of looking at it, but what it does is it tries to show you essentially sites that are close together on the graph are the most similar, and sites that are far apart are the most different. And these are our zones of interest. So each site is labeled here, in this case, by the zone of interest. And you can see there's a really big difference here between zone of interest 3 and zone of interest 2. And this is for carbon. So we've got a lot of geographical change across this corridor. And it strikes me it's been really difficult to produce models that account for all the variability, even just in this one corridor region. So it'd be very difficult to generalize even to other countries from this, if we can detect such big differences between just 2 regions on the same corridor. So that's the background. Unfortunately, it tells you which variables are most sort of significant in explaining, sorry, we're going a bit forward when I thought we were, which variables explain most of the variation. So we've kind of got 2 axes along here. We have above ground axes, so above ground carbon and saplings, canopy and saplings, and then on this axis we have below ground. So we've got these 2 neat dimensions. Essentially, the sites can all be well described by above and below ground variables. And you can label them by site. So you can see here that some sites are very similar, even although they have, and wait for it, very different land uses. So that site, we sampled 3 land uses very close to each other, and the site factor seems to be what's drawing them together more than the land use factor. This is just carbon. But we couldn't get significance out of that analysis, so we've done a principal components ordination, which ultimately what it does is a permutation analysis. And so it's very similar, sites which are close together. So green is closed canopy, and that's across a number of sites and zones of interest. And pink is reforested. Orange is tree fallow. So we can see general clusterings here, but it's a bit murky. It's a bit like reading tea leaves. If we do an ordination, a permanova, what we find is that the 2 explanatory factors here are actually site and land use. So site is really important in predicting carbon, and land use is also important. We can't test zone of interest yet because we've only put in the data from 2 zones of interest, but when the 3rd goes in, we should be able to actually test whether that's significant, and my guess is it will be. But there we go. For carbon, you could predict your carbon stock from the site and the land use. Now, at least land use is coming out as an important predictor. If we do the same thing for hydrology, the only thing that's important is site. And if we do the same thing for biodiversity, the only thing that's important is site, which is kind of not what we expected, although our data set is still sparse. So I suppose there's things missing in here, like we can put in and we will put in connectivity to the forest matrix for biodiversity, which is likely to be an explanatory factor. And time since clearance. Remember, at the moment, we've lumped together shrubs of oca that have been on the degraded pathway with shrubs of oca that are on the recovering pathway. So they could, although they look the same, be quite different in their biodiversity. So there's still things we have to separate out. But the final stage of this has been to try to relate these different data sets. So I've looked at the 3 things in isolation: carbon, hydrology, biodiversity, and what explains them. The next analysis is where you relate the similarities in 2 different matrices. So you compare the site similarities for carbon with the site similarities for biodiversity. And that starts to look at whether there's interactions or interrelationships between the 2. And so we essentially look at this number here in the hydrology similarity matrix and compare it using a rank correlation with that number in the biodiversity matrix. And here we do find, so this is a map that shows similarity from the biodiversity perspective with the vectors plotted that show us which carbon variables would help to explain the spread of that biodiversity data. And we find a weak correlation, but it's highly significant, between carbon and biodiversity. And we find if we fit models, that the best model is the most parsimonious model is just a 1-variable model, which I suppose it's not terribly surprising that soil organic content in the top 30 centimeters of soil explains the soil biodiversity sample that we took in the same hole, more or less. But for hydrology, we find, again, a weak but significant relationship. And here we find the best model for predicting biodiversity from hydrology would come from bulk density, which is how much a certain volume of soil weighs, so lighter or heavier per cubic meter. And that could relate to porosity. So a lighter soil might be more porous, but that's actually a different measure which didn't come up as significant. Maximum rooting depth came up as significant. And of course, in the literature, there's a lot of discussion about the importance of plants in terms of engineering soil space to create space for biodiversity. So that's not necessarily a very surprising result, that you need to have good active plant growth to open up the soil for biodiversity. But potentially we're also seeing that the biodiversity creates the carbon, because remember that strong correlation in the last graph. So I was trying to think about this in the wider context. There's obviously a lot more we can do to try to understand these relationships. But and it's encouraging to see that, for example, in the annual report, ESPA has flagged up that soils are really an area that needs more attention. So in the annual report, they said the importance of soils and soil processes is being recognized in a growing number of projects. So what we've done is quite correlative so far. It's a little bit mechanistic, especially when we managed to tease apart the site histories, which we're doing by interviewing local communities. Then we'll know if it's a degrading site or a recovering site. Then at that point it becomes much more mechanistic, our information. Now, this paper came up by Bill Sutherland, and it really got me thinking. People tend to think of biodiversity itself as a service, you know, that biodiversity is what we want out of these systems. But actually, I think it's really important to understand more how biodiversity creates carbon and how biodiversity creates the hydrological services. I don't think we should think of just biodiversity as a third thing with valuable existence value that does nothing. It's not just providing bushmeat, it's providing these other services, and we need to understand better how to facilitate it to do that. So we need to get a bit more functional. So the next steps is to boost this analysis up to 45 sites when all the lab work's done, and account for spatial autocorrelation by adding the third zone of interest, and we can do some more sophisticated spatial autocorrelation statistics as well. We need to understand the site history a bit better, whether it's on a recovery or a degradation pathway, and we can also go a lot further and explore the above and below ground relationships. So I've just shown you one strata of soil biodiversity, but we've also got canopy biodiversity. So we can look at the relationships in the canopy and then between the canopy and below. So I want to move on to my thank yous, and just to point out that this is a very hard won data set by a lot of very wet people who quite commonly got ill. And we did lose a motorbike in a river, and it was a bit of a struggle to get to these sites. So I think it's particularly amazing that they managed to get to the same hole in the ground on time and bring in these data sets. And also, thank you very much to the teams in the capital city who were constantly correcting and checking and double checking and entering data for it. So thank you to everybody.
Thanks, Alison. We have quite a bit of time for questions. Are there any questions?
Thank you, Jeff Fager from London International Development Centre. Alison, that was really interesting. I'm going to ask you a rather big question because I was interested in the link between your talk and Mafaka's talk earlier. So you're working on upland rice under Swidden agriculture. He's working on lowland rice under irrigated agriculture. And this is a policy-focused question, but it ends up with a natural science question. So in Africa today, it's likely that most, I think, agricultural policies over the next 20 years are going to focus on intensified rice production in lowland irrigated systems. Now, that's not appropriate for the communities that live in highland systems, but that's where the money is going and where the money probably will go. So an interesting question then, if that's the trend, it's certainly the trend in Madagascar with their program of rice intensification. If that's the trend, then it's an interesting question to ask whether, in terms of biodiversity conservation, it is you're getting more out of ecosystem service research in terms of biodiversity conservation in upland Swidden rice cultivation or lowland irrigated rice cultivation. And I'm wondering whether the projects we've heard about this morning have the capacity to compare with each other about biodiversity conservation, the other ecosystem services associated with those two alternative rice production systems, because I don't think the decision between them is going to be made environmentally over the next 20 years. It's going to be made by food security decisions to feed another 500 million Africans who are going to appear over that period.
I'll just get a few more questions and then you can do a batch answer.
I'll try and write them down.
Okay. Yeah, thanks, Alison. Two questions. One of them is, do you actually have a lead to your initial question on whether payments for ecosystem services can deliver poor goods and services, and especially emphasizing on sort of the poor aspect? I don't know whether you have data that's leading to that. It would be interesting to know how you see that. Second question is it's a bit surprising from your graphs that the biodiversity aspect is not really driven much by land use, but more by site. And I'm very keen to know maybe why that would be the case, because I think that goes quite against the grain of science, sort of. So maybe a bit of discussion on that.
Yep.
Yeah.
If you want to get those two, and we'll see.
Yeah. So first of all, yes, there's a very big difference between upland and lowland rice. Now, the hydrological sampling that the hydrology team have done has focused on whole catchments and the relationship. Now, the communities are very different. So the same people don't own the as the upland agriculture. The paddy system is much more stable. There's an inheritance system, and the upland agriculture is much more driven by illegal deforestation. So in Madagascar, Tavi agriculture, slash and burn, is illegal. And yet it's producing such a huge proportion of of productivity. And this is a bit of a problem because it has direct downstream effect, and the RED project is largely focusing on controlling the deforestation in the high part of the catchment, which is affecting the people in the low part of the catchment. So there's two very different communities affected in different ways. And that's different components of our project, and the socioeconomists are looking at that. And it's difficult because there's really no legal incentive or framework for trying to perhaps suggest useful interventions in the higher system, because people just seem to want it stopped. So if you could intervene perhaps to extend the fallow periods, you could actually make that quite a sustainable system, but people don't want to talk about that because it's illegal. So that's a bit of a problem at the moment.
I want to give a quick answer to Sam because we've just run out of time.
Below ground could be quite different to above ground is one reason why we might not be getting what we expect. But of course the other reason is the history. So we haven't tangled whether a site is newly deforested or been deforested a long time. And then of course the connectivity to contiguous forest could be a bigger explanation than the land use. You know, if it's right next to good quality forest. So that those, sorry.
Thanks, Alison.